So thank you very much, Martin, and it's indeed a great pleasure to be here. And what I'm going to tell you about today is a cautionary tale with what I think is a happy ending. So we want to focus a little bit on what is predictive toxicology supposed to do, and what it should do is enable us to more easily screen the many, many chemicals that we have to consider, and ideally without using animals, but I think that's a bit unrealistic. I think I need to tell you that in order to do the prediction correctly, you need accurate assays. If the assays aren't accurate, then neither will the predictions be accurate, and that's a problem. Current models and approaches work to some degree, but how well they work depends on how high you set the bar. So how stringent are your criteria for success or failure determines whether it worked or not. What are called predictive models for most chemicals perform very poorly. I'm going to tell you about predictive models for obesogens, chemicals that cause fat cells to differentiate or that cause animals to become fat or obese. Those work very poorly. And I think the accuracy can be improved by adding additional screens. We talk a lot about high throughput assays. So tests we can do many, many tests in one time. And I have a colleague who's very fond of saying, I can get you the wrong answer very quickly. Is that what you want? And you need to keep that in mind because that's a lot of what we're getting. And I think the message is that predictive toxicology must be grounded in biology. The predictions that you make have to be borne out by the observations that you make in vivo. You cannot have them separately. Okay, so you probably know about in the United States, we have what was called the Food Quality Protection Act that directed the Environmental Protection Agency to develop a screening program using properly validated assays to determine whether there were endocrine active substances in food and in water. And in 13 years, they finally had a few assays that they were ready to do, and the screening started. And the EPA's approach to assess chemical risk is you have a bunch of chemicals, and we do some chemical screening, these high throughput assays, which I told you about ToxCast. Then we need to know if they're exposure, because hazard is fine, but without exposure, you can't calculate what risk is, and we'll hear about that later. Prioritization of chemicals for more deep testing, and eventually trying to integrate this with many, many years of animal toxicity studies to try to see which of these assays make sense to predict effects in humans. So here's ToxCast. You have chemicals. You have a bunch of assays. Probably it's more than 500 different assays now. You carry the database of chemicals. Is there a hazard? If there is, then maybe you want to prioritize the chemical. Initially, ToxCast was conceived to do risk assessment. And the risk assessors said, no way, we're not going to do that. So ToxCast was reinvented as a prioritization tool. So to decide which chemicals should go into detailed risk assessment. And ToxCast has kind of morphed into what's called Tox 21 now, toxicology for the 21st century. And the goal is to screen around 10,000 compounds with a large number of assays to get a reasonable idea about which ones might have health effects. And the EPA now says that this approach is good enough to prioritize estrogens for screening. And the people I know in the endocrine disruptor community would not agree with that. And to the EPA, estrogen is defined as active in 2 out of 18 estrogenicity assays. But these assays include receptor activation assays, receptor antagonism assays, and ligand binding assays. So to a nuclear receptor biologist, which I am, you need to do better than 2 out of 18. And surprisingly, they didn't feel the need to consult experts in estrogen signaling. So you know the EPA is in Research Triangle Park in North Carolina, and 10 minutes away is the world expert on estrogen receptors, Ken Korak, and they never asked him. So that's a little bit of a cautionary tale too. So how did I get involved in this? So as I said, I'm a nuclear hormone receptor biologist. I identified a number of hormone receptors. I identified ligands for hormone receptors, developed the methods to do this in the early 90s. So we asked the question, because this receptor PPAR gamma is a key regulator of fat cell development, and ToxCast had PPAR gamma assays, 3 different assays that they used, so we asked the question, or we were induced to ask the question by the National Toxicology Program, are these results valid? So PPAR gamma is a receptor I actually cloned, and it's my patent, so I know a little bit about that receptor. So we asked the question, do the assays work Do the high throughput assays correlate with what you see in the lab? And do they predict whether the chemicals will make cells become fat cells? So we tested the activity on the receptor, and we tested their ability to make fat cells in 2 different cell culture models. So here's what we were given. This is a list of the top 20 ToxCast activators. And when we tested these, we found that they weren't all active. So you can see the ones that were active, there were just about 5 of them. Surprisingly, some of them were actually antagonists and many were inactive. So Pete Myers brokered a meeting with the EPA guys, and we sat down and we talked with them. And they said, they looked at me in the eye and they said, if you knew what we knew, you'd realize how wrong you are. I thought, okay, that's interesting. So we went ahead, we tested them more, and we also tested them in adipogenesis assays. So at some point down the road, I called Richard Judson, and he said, well, you know, the list you have is an old list. We've changed it. And he said, we think that these chemicals are very likely to be active, and these chemicals are very likely to be inactive. So that's a little bit better, right? So you see that many of the ones they thought were active are active, and many of the ones you thought were inactive are inactive. So that's improved. The ones that we showed were active all induced cells to become fat cells, and the ones that were inactive did not. So no surprise. And here's just an example of the way the assays look. Here's the positive control compound, which is a pharmaceutical chemical called rosiglitazone, otherwise known as Avandia. And you can see that the chemical triphenyltin is much more potent than that, and the other chemicals are much less potent. Here are the antagonism assays. Here is a pharmaceutical antagonist, and these are the chemical antagonists. So the assays are reasonably robust. So the most favorable interpretation of this data is that 5 out of the 16 chemicals that they tested as actives were actually active. 3 out of the tested actives are antagonists, so have the opposite effect, and half of them were inactive. So one question is, why are so many of these top 16 tested, not predicted, tested active? Why are they inactive or antagonists? That's a kind of a problem. And I think the answer is that the assays were substandard. So we'll get to that in a minute. This title is kind of funny. The last time I gave this talk was in Barcelona. So this says, in Catalan, how tasty is a tox pie? Okay, so in 2011, there was a meeting at NIEHS that was put on by the National Toxicology Program, and a variety of groups were invited to nominate assays that predicted adipogenesis, feeding behavior in 2 species, insulin sensitivity, beta cell function, and islet cell function. So we nominated assays. David Reif, who was then at the EPA, generated what were called tox pies, or toxicological priority indexes, for each of these endpoints, and he picked chemicals that were either predicted to be quite active, predicted to be medium active, or predicted to be inactive, not in just one, but in all of them together. So the ones that are predicted to be inactive are negative in all the assays. So the recipients, including us, received the chemicals from the National Toxicology Program. We started testing these. So this involved many people in the laboratory over almost 5 years. Unknown to us, the predictive models changed many times, more than 10 times over that time period. So it was a moving target. We didn't get new chemicals each time, just the models changed, and no one told us that. So what happened at the end of the day was the study became unpublishable by most of the people because the models that were being used were different than the chemicals that were tested. But we didn't accept that because we invested a lot of effort into that, so we wanted to know, what did we find? So here's the tox pie. It looks like pie, so each one of these assays represents a slice in the pie, and I won't go through all of them, but some of these are nuclear receptor assays, some of them are other cellular signaling factors, and this one here is supposed to be an assay in which the chemicals were inactive, but we found out later that it couldn't do that, so we had to leave that one out. So here's how it looks. The highly active chemicals are over here, the medium active chemicals are over here, and the inactive ones are over here. So the NTP gave the chemicals to the 7 labs involved, and we tested them. Here's how the chemicals look. These are the tox pies, and you can see the size of the slice reflects how active they are in the assays. And I'll, as we say, cut to the chase and show you these are the ones that were active, so it's not all. I'll show you actually some of the data. There were some chemicals that we did not know already were active, so here's the receptor PPAR gamma. This is an activation assay. Here's rosiglitazone, here's rosiglitazone, here's triphenyltin, And here are two chemicals that we didn't know before were active. And here's another receptor called RXR, which is the heterodimeric partner of PPAR gamma. And you can see that here's the pharmacological activator, here's triphenyltin, and here's one of the chemicals that was active. So only a very small number of the chemicals that were tested to be active were actually active in our experiments. So this shows induction of adipogenesis, so treating cells and causing them to become fat cells in culture. And you can see here are the positive controls here, and these are 4 different doses of the test chemicals. And you can see that most of them induce adipogenesis to a strong degree, some to a lesser degree. And these two chemicals, this is the PPAR gamma activator, this is the RXR activator, and these other chemicals act through a different pathway. We don't know what that pathway is, but we know what it's not. Here's gene expression in the same assays, and you need to see that these 3 fat-specific genes are induced to have a normal functional fat cell, and you can see that these guys do. The other chemicals induce the expression of these genes to various degrees. At the time, that was difficult for us to interpret, but since then, we've learned that many of the chemicals that induce cells to become fat cells make abnormal fat cells that don't respond correctly. So that's one of the problems with this class of chemical, that they make fat cells that store fat, but they don't function in other ways that fat cells should. Here's again the 2 known activators. This is in the stem cell differentiation assay. Only 2 chemicals were active, the PPAR gamma activator and the RXR activator. They promote adipogenesis and they promote appropriate gene expression. So these make functional fat cells. This summarizes the results. It's a big table, you don't need to see it, but you can see that less than half, about 40% of the predicted actives were actually active in the adipogenesis assay. Unfortunately, 2 out of the 17 predicted negatives were also active. One of the predicted actives was actually an inhibitor of adipogenesis, which you wouldn't expect. And 2 out of 7, I already said that, 2 out of the 7 predicted negatives induced adipogenesis. Okay, so at the same time, poor Scott Auerbach had to publish the paper with all the rest of the groups trying to summarize what they learned. And because they couldn't test anything, because the model changed, this was published as a review article. And we declined to participate in this paper. So to summarize what came from this prioritization and what we showed, they tested actually in silico 1,890 ToxCast Phase II. So the second generation of assays we tested, ours came from ToxCast Phase I. They ranked those chemicals from 1 to 810 based on how active they were and using a variety of metrics that they were happy with. And the bin 810 actually had 1,000 chemicals that were predicted to be inactive. So a score of 810 is a very large number of chemicals. And they prioritized 30 chemicals for each one of the assays that I told you about, each one of the endpoints. So 2 for adipogenesis, 2 for feeding behavior, etc. Interestingly, none of the chemicals that we tested and showed to be active were on their prioritized list. And 3 chemicals were on both lists, on the initial list and on the subsequent better list, and those were all inactive chemicals. The prioritized list had some chemicals that just looking at are going to be active. Tetrabutyltin, tributyltin benzoate, farglitazar, these are known PPAR gamma activators. But there's some obvious inactives. I guarantee you acrylamide's not active, aspirin's not active, the 3 inactive chemicals are not active. So the list could be better, let's say. And no testing was performed in the Auerbach paper because all of the people involved had the list of chemicals changed so many times that what they did was not usable. The chemicals that we tested active when we compared them on the list ranked number 48, 71. This is triphenyltin, the one that I showed you was more active than the positive control, 96, 150, all the way down. So they were all over the place. So there was really no good relationship between the chemicals score in the predictive model and what we found using the model in cells. So what is the source of the problem? And I'm a nuclear receptor guy, and I can tell you the problem is the assays. Why do I say that? The best evidence is this. So these are the ToxCast Phase I on the bottom and Phase II assays conducted in the same laboratory on the same chemicals. And you can see that for PPAR gamma, the R squared is only 0.4. So that's not significant. For the ligand binding assay, it's only 0.18. So that says there is a problem with the assays because they're not reproducible even by the people who did them the first time. And I can tell you, in my laboratory, if you can't reproduce your work, you get to go work somewhere else. You don't get paid for that. Another thing that EPA is untroubled by is the lack of congruence between the assays. So this is PPAR gamma activation. This is their main activation assay, comes from a company called Adipogenesis. This is ligand binding, and this is Tox21, which was done by the National Toxicology Program. So ligand binding and receptor activation have to be 100% congruent, right? You can't activate the receptor if you don't bind. Or if you antagonize the receptor, you better also bind it. So they're completely untroubled by the fact that there's a very small overlap between binding and activation or binding and antagonism. They're also untroubled by the fact that a large number of their activation assays are more like Tox21 antagonist assays. So to a nuclear receptor person, that is a big red flag. To the EPA, it's, yeah, that's how it is. Despite that, we learned something interesting from this exercise, and that is that many fungicides are either obesogens in vivo or they're potential obesogens. So here are some: tributyltin, dibutyltin, triflumazole, polyfluoride. These are chemicals that are tested to cause animals to become fat. And it says there which receptors are active. These are almost all PPAR gamma activators. Here's two that we came from the testing, quinoxafen and zoxamide. They make cells in culture become fat cells. That means they probably are obesogens, but no one's tested them yet. So to me, in order to be a bona fide obesogen, you have to show it in vivo. In cells or in silico is not enough. This chemical, fludioxanil, activates RXR, so that's likely to also be an obesogen, but someone needs to test it. And this chemical, flucilazole, we don't know what pathway it activates, but it causes cells to become fat cells. So from this exercise, despite that it was imperfect, we learned a fair bit about what chemicals might be obesogens. So I beat up EPA a lot, but I would like to say that it's possible to do these assays better. So this paper came from Chad Dyson-Roth's lab. He was formerly at the Hammer Institute. So they worked with the EPA and they prioritized chemicals from ToxCast assays. They had 49 predicted activators, 11 predicted antagonists. They tested them on 7 different kinds of assays and asked the question, how well do the predictions work? And the answer is 29% of the predicted activators worked. So that's either good or bad, depending on your perspective. I think that 29% is not adequate. I think they're happy with it. So one of the smarter things that EPA ever did is they hired Chad Dyson-Roth to run those assays now for them. So lastly, Jane asked this question, and Cassini also asked me this question. There seems to be this idea out there that in order to be an obesogen, you have to activate PPAR gamma. So here's PPAR gamma, and it is the main regulator of whether a cell goes from a pre-adipocyte to an adipocyte. And it does that by regulating the expression of quite a few genes that are characteristic of adipocytes. And there's this trend these days, we want to do what are called adverse outcome pathways. We want to construct cellular pathways that we can use to assess whether a chemical is likely to have an effect. You can see that PPAR gamma takes input from a lot of other pathways. So it's regulated by CEBP, and it regulates CEBP. It's regulated by BMP signaling. It's regulated by insulin signaling. It's regulated by the presence or absence of its cofactors. There are many, many factors. So there is not one linear pathway. There are many pathways feeding into PPAR gamma signaling and which it also regulates. This also doesn't address the fact that we showed that RXR activation is important for making a pre-adipocyte. So you can't get a fat cell till you get a pre-fat cell. And the key player here is RXR. and we just published a few months ago, that when you make a pre-adipocyte with RXR and turn it into an adipocyte, it's a dysfunctional adipocyte. So that means something else is going on. It's making an imperfectly functioning adipocyte. So to summarize, because my time's almost up, I think that screening for the ability to activate RXR and PPAR gamma does a halfway decent job of identifying potential obesogens. However, that depends on the quality of the assays. You have to set reasonable standards and expect that the person doing the assays will adhere to those. And I also want to make the point that many obesogens, either potential obesogens or known obesogens, do not activate PPAR gamma. So do not accept that as the type of assay that has to be done. For our experience, combination of activation assays and adipogenesis assays in vitro is a reliable predictor of in vivo activity, at least for n equals 4. I think this also shows that there are quite a number of obesogens that haven't yet been identified. We've only screened a few hundred chemicals and we already know about 50. So I think that says there's probably some hundreds more. The ToxCast assays for RXR and PPAR gamma, I've told them many times, they don't care, are highly suspect. They don't work very well. And you can't get a good outcome if you don't start with a good input. The old-time computer programmers used to say garbage in, garbage out. And that's the same thing. If you start with inaccurate assays, you will get inaccurate models. It cannot be otherwise. The main problem is the uncritical reliance on assays and then trying to massage the data afterwards by post facto modeling to try to make it more accurate. You need to do it correctly in the beginning, and if you do that, it will work. So 21st century toxicology, so-called, it's very promising, it will work, but at the moment it's not working as well as it could. So here's the happy ending, I think, and you probably already know about this. The European Union issued a Horizon 2020 call for grants that will generate robust, reproducible, validated, and standardized assay for endocrine disruptor testing that can become OECD guideline assays. That, I think, is of critical importance. Looks like they're going to fund 8 consortia of many universities and a few governmental and non-governmental organizations. Here's a partial list, or this is actually the whole list. 3 of these actually assess metabolic endpoints, and I'm participating in one of them. So at the end of the day, because the European Union is funding the experts in developing the assays, they will get assays that very probably accurately predict biological endpoints. So I think this is promising, and I congratulate the European Union for their foresight in doing this. And that's all I have to say. I'm happy to take any questions that you might have.